Good morning, this is Eleonora Leucci from the Laboratory for RNA Cancer Biology at KU Leuven, Belgium. By mining publicly available data, we identified a cancer-specific transcript, which we now call LISRR, for long non-coding ISR regulator, whose expression and copy number is able to discriminate between responders and non-responders to immune checkpoint blockade in melanoma. So the transcript is able to recruit specific factors at the translational machinery, and these factors allow cancer cells to survive stressors, in this case the therapy, by assembling ribosomes at the endoplasmic reticulum. Translation of the endoplasmic reticulum leads to the production of specific proteins implicated in immunosuppression. Specifically, we found that LISRR allow for increased expression of the checkpoint inhibitor PD-L1, for the block of translation of potential neoantigen, and for the increase in the build of the glycocalyx or around the cancer cells. So the glycocalyx is sugar coat, which allowed the cells to escape the immune system. By using co-culture systems, in vivo humanized models, and patient explant, we also show that inhibition of LISRR increase survival and re-sensitize to immune checkpoint blockade. We believe our findings are intriguing because a large portion of melanoma patients still do not respond to immunotherapies. Furthermore, LISRR is not only expressed in melanoma but also in other cancer types, and thus it could be a viable target. Our results highlight also a novel mechanism by which a transcript can lead to the emergence of drug-resistant clones within a tumor. And lastly, we show that by targeting this transcript, we can specifically erase the glycocalyx, which is a hallmark of cancer, on cancer cells, leaving normal cells which rely on this structure for their function unharmed. And if you want to know more, read our paper. Enjoy your reading.